Testing, testing, testing.
Thanks, Jackie.
Good morning.
Thanks, Jackie.
Good morning, everyone. I'm Jackie, and today I'll be presenting on the specificity of gut germinal center derived antibodies to commensal gut bacteria. So essentially, my research examines the interaction between antibodies and the gut bacteria. And my research kind of bridges the two fields of microbiology and immunology. So I will be giving you some background on both. So gut bacteria and probiotics are hot topics in recent years. And you can find so many different types of probiotics products in the supermarket, from yogurt to kombucha to supplements. Um, but this concept of using bacteria to promote health isn't exactly a new thing. It was suggested by the Nobel laureate Elie Mekanoff in 1907. And since then, especially in the past few decades, a large body of research on gut bacteria has emerged. So our gut bacteria, or gut microbiome, which is the collection of gut bacteria in our intestines, is not a random assortment of bacteria that we eat. It is actually a stable ecosystem of bacteria that is resistant to change. As shown in the diagram on the left, gut bacteria inhabit or colonize the mucus layer, which is in green, and the bacteria are in pink. Um, and they live, and the mucus layer is directly on top of the intestinal epithelial cells. And the healthy gut microbiome plays a crucial role in maintaining our health. It helps us both absorb nutrients, trains our immune system, and also protects us from pathogens and even regulates our circadian rhythm. And we still don't really know the full extent of its importance to our health. And when our gut microbiome equilibrium is disrupted, it is correlated with many diseases, including diabetes, obesity, inflammatory bowel disease, and heart disease, and so many more. And since the gut microbiome plays such an important role in our health and in disease states, it is important to better understand how it interacts with its environment in the gut. And this brings me to the next topic of the immune system, since the majority of the immune cells in our bodies are found in our guts. So here's an antibody. An antibody is the fundamental building block of the immune system. They can neutralize harmful foreign substances, and they can signal the macrophages to engulf them. And the blue part on the bottom, or like the arm of the or the stem of the Y shape, is the Fc region, or the constant region, and its structure is the same in all the antibodies in your body, in all the antibodies of the same class in your body. And the red part is the FAB region, which is the antigen binding fragment. So this is the part that actually binds and interacts with the antigen usually. And the green part is the variable region, and its structure varies greatly across the different antibodies, and it is complementary to the antigen that it is binding to. So this is the antigen, and this is how we would usually bind to a protein. So these antibodies are usually produced in B cells, which mature in structures called germinal centers, and they're located in our lymph nodes, our Peyer's patches, and other secondary lymphoid organs. So in the dark zone, the B cells start off in there, and they multiply and they mutate. And then they migrate to the light zone, where they interact with other immune cells. And the B cells that produce antibodies that can bind really well to the target antigen will be given this survival signal, and they will migrate back into the dark zone and go through the entire cycle again. And those that can't bind well to antigens undergo apoptosis. So at the end, the entire germinal center will be filled with B cells whose antibodies have a very high affinity to a target antigen. So the gap in knowledge is that this process that I just described, the B cell maturation, is based on the immune system's reaction to pathogens or bacteria or viruses that are harmful to our body. But we don't know anything about how this process applies to the immune interaction with the beneficial gut bacteria. So this leads me to my research question, which is, what is the specificity of gut germinal center derived antibodies to common commensal bacteria species? So in other words, why were these B cells selected in the germinal centers? Or like, why did they win? So here are the materials and methods, and here's an overview. So we had 3 different types of mice, which I will introduce later. And from each mouse, we would extract the germinal centers from the gut lymphatic tissue, or usually the Peyer's patches or lymph nodes in the gut. And from that, we would extract the antibodies, and the 6 antibodies that we used in the study are as shown in the blue box on the right. We also had different bacteria isolates, so we had different cultures of specific species and strains of bacteria. And we also extracted the cecum, which is a structure in between the large and small intestine of the mice. And we kind of used that, we used the cecum because it has an intermediary composition of bacteria that is kind of in between what is in the small intestine and what is in the large intestine. And we ran these through dot blots and Western blots, and the Western blot separates the proteins in the sample via size, and the dot blots are just, each individual dot corresponds to one specific bacteria species because it has so we used 3 different type of mouse models, and the first one of bacteria that the germ-free mouse. Kind of in between, so it is grown, it was born in, and it was raised in a sterile environment, which means that there is absolutely no bacteria in them. Dot blots and Western blots, and the second type, the Western blot, oligo mouse microbiota, or MM12 for short, in the sample, and it is also raised in a sterile environment, but it has 12 specific types of bacteria in them. So it's a very controlled population of bacteria. And the third is a specific pathogen-free, or SPF mice. They are essentially healthy microbiome. So just an overview of how some of the results are presented. This is an example slide of a Western blot. And as shown, the leftmost column is a germ-free mouse. The middle column is MM12 mouse, and the SPF is the third most column. So since the antibodies were just isolated from the gut without knowing, we don't know what antigens that these antibodies bind to. So we had to determine whether these 6 antibodies that we isolated bind to bacteria present in the gut. And to do this, we ran a Western blot of fecal contents of germ-free MM12 and SPF mice using the 6 antibodies isolated from the gut. And as I explained earlier, in each slide or in each membrane, the leftmost column is germ-free, the middle is MM12, and the right column is the SPF fecal samples. And as shown here, so there were 3 antibodies that showed binding to these membranes, and 3 that didn't. And we know that these 3, or we hypothesized that these 3 antibodies can bind to bacteria in our guts, or in the mouse's gut, because there was no binding in the germ-free, which means that because there's no bacteria in them, there's no binding, but there was binding in MM12 and SPF mice. So using the 3 antibodies that did bind to the bacteria content in the mice, we then ran a dot blot using these 3 antibodies. So it's kind of hard to see, it's kind of hard to see, but in the 3 panels, there are a bunch of different bacterial species that are each represented by a single dot on the screen. And we found that all 3 of these antibodies bound to the same 3 bacteria. And this happens to be the Lactobacillus johnsonii, the Staphylococcus aureus, and the Proteus mirabilis. And this result wasn't expected because the 3 antibodies should bind to different antigens on different bacteria. So this kind of made us question the validity